Good afternoon. Good afternoon. There we go. Good afternoon, everybody. Y'all were in, but you weren't in. Good afternoon. Good afternoon. We're still missing quite a few people. All right. I missed working with you all on Thursday, but hopefully you all worked on your worksheets and completed. Let me go on back over there and see if. All right, so hopefully you completed that worksheet. Let me see if I can pull up mine that has some answers on it. Because that's what you were supposed to be working on Thursday in my absence. So hopefully you did that. I'll stop sharing for a minute and share my page. It has some of the answers on it. I'll quickly go over this and then we'll do today's because like I said, you should have. I worked on this with you, got it started the other day, and hopefully you were paying attention so that you were able to finish up working on this worksheet. Because this is due, this is due, this is due. All right, so those are the answers for the first three questions. And while we're waiting for people to come in, if you don't have those, check your paper and make sure that you at least have that much. All right, so I'll at least look at that. I'll review it quickly when most people are in for the time being. Let this be the warm-up activity to make sure, because this is due today. It'll be late after today. So if you haven't done it, you need to be working on it right now. Okay. You got half the class here. Half the class. Half the class. Folks are good and late today. I give another 2 minutes. Another 2 minutes. Well, I'll do attendance. What happens when I mark them absent and then they show up? Good afternoon. All right, give me a second. I'm gonna come back to that, folks. Someone asked a question, and I'm sure you want to know the answer to this too. All right. All right, the other worksheet that you had to turn in was this one. And the boxes at the bottom are similar to what we were doing with today's worksheet. So completing this, and let me see if I have, give me a second. Let me see if I can't pull up my copy so that I can actually write on it. Oh, here come all the late folks now. Just give me 1 second. If I pull mine up. Y'all are late. Good afternoon. Trying to make sure that the last week's paper is turned in, and then we're moving on to incomplete types of inheritance. So please use this next 5 minutes to make sure last week's worksheet has been turned in. All right, give me 1 second to pull up my copy of that. That way I can write on it. Maybe. I don't think so though. No, because this is a PDF. Okay. All right, so it doesn't matter. Whoops. Okay. We're gonna come back to that in a minute. All right. So I'm gonna quickly go through this in case you haven't, and I can't write on it, so you're gonna have to follow me. But this is a Punnett square, and the Punnett square again represents the four possible results that you can get when you take the egg or sperm of the mom and dad, and they can create four possibilities. Okay, so these letters on the top and the side represent and we can call the top dad and we call the side mom. In this case, we're talking about plants. So they're not giving you male and female, they're just telling you what the two parents are. Okay. Okay. All right. In the worksheets that we were working on, and hold on, folks, people are coming in late, so I'm having to stop to let them in. All right. So again, and actually I could use today's worksheet to do this as well. But anyway, the letters at the top represent the gametes, and we said during meiosis. And folks, we are reviewing also, so consider this review because you have a midterm exam. Your class is next Monday. March the 2nd is a 5th period midterm exam. So check your webpage, and I'll show it to you in a minute when we finish doing this for all of your midterm exams, but the ones for 5th period are on March the 2nd. So we will be covering cell reproduction and genetics. Okay, because meiosis and mitosis has everything to do with genetics. So please be aware of that. All right, so again, during meiosis, not mitosis. During meiosis, we can have the separation of the alleles, and these are alleles, to where the mom can give this one to this box and dad can give this one to that box, and we get the big B, little b. Or mom can give this one, and y'all watching my arrow, they can give this one and this one, and then we end up in this box with another big B, little b. Or we can take the top and this one and that one and put that in this box, and again we get big B, little b, and this one and that one, and we get big B, little b. So in this particular case, all of them are going to be the same, where you had two, and this is a rule. When you have 100% dominant mated with 100% recessive, you're going to get 100% heterozygous dominant. Okay? So that's a rule. That's something that you can remember. 100% dominant cross with 100% recessive always gives you 100% heterozygous for whatever that dominant trait is. So whatever B represents in this particular case, that is what all the offspring will look like. Okay, so we've drawn the genotype. And again, this is all review, folks. So hopefully you're paying attention. Even if you've claimed you've turned your worksheet in, pay attention. This is review. This is what we've drawn in the box is the genotype. So we know what genes are present. And then we have to take that information, and I just said they're all 100% dominant. That's the phenotype. What do they look like? They're all going to look the same, and they're all going to look like the big B individual because that is dominant. The capital letter means they're dominant. All right, in the next one, we have two heterozygous individuals. So it's the same thing as if we took this generation and we said, I'm going to mate the two people from this one, the big B, little b. So in this case, they took big T, little t, and both parents are big T, little t. So they're both heterozygous. So in this case, and in this case, you're always going to get the same thing as well. In the first box, you're going to have two capital T's. In the second box, you're going to have a capital T, short t. And the same, they did one for you over here. All right. And then this last box, you're going to have two small t's. So as a rule, your 100%, and every box represents 25%. As a rule, whenever you cross a heterozygous with a heterozygous, you're going to get one homozygous dominant, two heterozygous dominant, and one recessive. Okay, always, always, always. And I say this because you can, when we take the test next week, those are rules, and that's a question that they will ask. You don't even have to, you can draw the Punnett square so that you can back yourself up, because this is like testing, proving your answer. But if you remember this, you can just answer it off the top of your head. Dominant plus recessive gives you 100% heterozygous. A heterozygous with a heterozygous is going to give you one dominant, two heterozygous, and one recessive. Okay. Next box. You had capital A, and capital A goes in this first box. You're going to have a capital A and a short a in this second box. Capital A over here, and A, capital A, small a over here. So you have 50-50 on this one. However, even though you have 50-50 as far as genes are concerned, and this is where it gets tricky. If they ask you the genotype, these are all going to be, except for this one, I'm sorry. The genotype is going to be, sorry, not except. These first two are going to be capital A, capital A. They're going to be homozygous dominant. Even though this one was heterozygous, they are producing homozygous dominance 50% of the time. The other 50% of the time, they are producing heterozygous dominant individuals. And I say that because we don't know what these letters represent. Okay, so you do that with all of these. This one, the X, the Y's, and this is probably yellow because this is not to be confused with the meiosis where we have male X and Y individuals. Okay, so this Y is obviously like for yellow and the other green being recessive. So that's that one, the same thing with X over here. For some reason they put whatever characteristic, but this is going to be not the sex cells. Okay, so let me do this one over here. They give you the information in the box, and you had to say, in order for me to have two Y's here, there had to be a capital Y here. There had to be a capital Y up there. In order for these two to have a lowercase y, they had to get it from somewhere. So if we already know this one's a capital Y, then that means that this one up here had to be the lowercase y. Okay? If that's a capital Y and this one got a capital Y, it got it from this one. So these two are going to be capital Y's. Up here, you're going to have a capital Y and a lowercase y. Based on because the only way you can get those results in these boxes down here is if there was a lowercase y here. Everybody else is a capital Y. Same thing with this box. The only way that you can get a lowercase e in this box is if there was a lowercase e here. Everybody else had to be a capital E. All right, this one's easy because everybody's the same. So you're going to get lowercase n's in all four boxes. All right, this one, in order to have one X that's capital, you had to have at least an X, capital X here, and a lowercase x there. Okay. If you have two lowercase x's here, that means this one also had to be lowercase, and that one has to be lowercase, because in order to get this box, you're getting it from this box here, this corner box, and this corner box is producing this one. So both of these have to be a lowercase x. And for this one to have a lowercase x, it also had to be a lowercase x here. So the only capital X is going to be in this first box up here, which means this box down here will be a capital X, lowercase x. This will be two lowercase x's. Okay. All right. I think I got all the late people now. We're still missing some folks. All right. So Jayla, I hope that was helpful. All right. And I hope for those of you who had not turned that worksheet in, you were paying attention. All right. So last week we did this one, the worksheet that you were supposed to work on your own was this one. And this one required that you not only fill out the boxes. And again, let me share mine because mine has answers on it. So I can do all the answers. I just did some for demonstration. You need to have completed the rest. And shame on you if you have not. I think I pulled up my answers. Okay. And it also helps to color code it. All right, so again, when you're doing these Punnett squares, it's not only enough to be able to make the Punnett square, but you have to be able to read it. So there are 5 things that this box can tell you. And again, all the problems are word problems, so you need to know your vocabulary so that you can properly draw what they're asking you to draw. So again, the first one said to you that in a pea plant, you're going to use the letter T for tall and the capital lowercase t for short. And again, you are not using two different letters. Whatever the dominant trait is, you use the capital letter for that. And the recessive trait is going to use the lowercase letter for the dominant trait. Tall is dominant, we use capital T. Short is recessive, we're using a lowercase t. All right, so it tells you. All right, so that's the setup. Then the question, the actual cross is a heterozygous tall plant. Heterozygous means that they are not, the alleles are not the same. You have two alleles for every trait. The alleles are not the same if they're heterozygous. So that tells me big T, little t. It tells me that. And I'm saying this, I'm repeating this because on the test that you're going to have next week, it's going to ask you to, what does that explain to you? Okay? So I hope you're paying attention because some of y'all are playing games while people are talking to you. All right, you're crossing, so it says you're going to cross a heterozygous tall plant with a short plant. The only way the plant can be short is if it has both genes for shortness. Okay? So that tells you if it's short, it has a little t, little t. It tells you that. Okay, you don't have to go, how am I supposed to figure that out? You need to understand the only way that the recessive trait shows up, you got to have both alleles. All right, the gametes, they wanted to know the gametes. We said we're just going to say that we put them in our Punnett square. So the parents, the gametes for each individual will contain, one parent's gametes are going to contain big T, little t. The other parent's gametes are little t, little t. Those are your gametes. So if you drew your box, that's the gamete part of the answer. Then you had to fill in the box. It said complete the Punnett square. So then you had to take the letter from the top and the letter from the side, put it in the box, top, side, put it in that box, top, side in this one, top, side in that, and the one on the bottom. Now we filled in the box. We got to figure out, what does the box tell me? The box tells me, I'm looking here, there are two of them are big T, little t. So that's two heterozygous tall individuals, and two of them are big T, I'm sorry, little t, little t. That's two homozygous. Homo means the same, the letters are the same. Zygous short. So a ratio is 2 to 2. Remember, ratios are always the, math ratios are numbers. So even though I've described it with words, which is what you need to do, you need to do this first before you can give me the numbers. All right, then they want to know phenotype. So this tells me what genes are present. This tells me what they look like. All right, so we leave out the words heterozygous and homozygous, and we just say, when I look at this plant, it's tall. When I look at the plant, it's short. So there are two of them that will be tall and two that will be short. So again, 2 to 2. All right, over here. The guinea pig's fur, black fur is dominant, white fur is recessive. So they tell you that they're crossing two heterozygous black guinea pigs. Okay, so that's telling you, if they're heterozygous, that the genes for those individuals are capital B and a lowercase b. Heterozygous means the two alleles are not the same. And both parents, you're crossing two parents that look like that. So big B, little b is crossing with big B, little b. And you do need to put that X in the middle. You do need to say, I'm crossing this one with this one. Okay, so you didn't put an X there, you're not completing that sentence correctly. All right, so then I put my gametes in the box, two up the side, one parent at the top, one parent on the side, and then I fill in the boxes. Big B in that box, little b, big B in this box, little b, big B in here, and two little b's over here. Then I have to read my boxes. So in this box, both B's are capital, so they are homozygous black-furred guinea pigs. These two are capital B, little b. So there's two of them that are heterozygous, but heterozygous black fur because the B, capital B is dominant. So they're going to have black fur. And this one, two small b's is recessive. So there's one homozygous white furred guinea pig. So our genotype is 1 to 2 to 1. And hopefully you wrote your ratio, because if it asks you for a ratio, you need to write it like a ratio, 1 to 2 to 1. Now, phenotype is different. These three are black. So what do they look like? We're not worried about homozygous, heterozygous. What do they look like? Three of them are black furred. One of them has white fur. So it's 3 to 1. So over here, the genotype and phenotype are the same. Over here, they're not. So it's not always going to be the same. And this one is a little different also. This one, you are attached earlobes are dominant to free hanging earlobes, and you are crossing a homozygous dominant individual. So that's our capital A, and with a heterozygous individual, that is a capital A, lowercase a. So one parent, the dominant one is going across the top, the heterozygous one is coming on the side. We fill in all the boxes, big A, big A, big A, big A, big A, little a, and big A, little a. And then we write it out on the side, but we see you got two homozygous attached earlobes, and then you have two heterozygous attached earlobes. Genotype. So again, 2 to 2, because there's 2 and 2 over here. Now our phenotype is going to be different than the other ones. They're all going to have attached earlobes. So we don't say 4 to 4, we say 100%. Okay, 100%. All four boxes, mean each of these is worth 25%. 100% attached earlobe. That's what they're going to look like. Even though they have different genotypes, their phenotype, all of them will look that way. All right. So hopefully you finished the rest of it. That was what you were supposed to be working on Thursday. And I know some of you just took it like a day off, but that's on you. But it's due today. That's due today. All right. We are moving on to, because I know a lot of you just take a holiday every time you want to take a holiday. So we are going on to types of inheritance. So I have to switch screens. So again, if you're not paying attention, that's on you. We do have a test next week, and the test will cover this. All right, so that's how. So we are going to finish types of inheritance and pedigree before the test next week. So make sure you're present. Make sure you're not late, because like half of y'all were late this one and this afternoon, and that kind of holds things up a little bit. All right, so we're going to look at 4 different types of inheritance: complete dominance, incomplete dominance, codominance, and sex-linked. Okay. All right, so complete dominance, and I'm going to go through this quickly, but I also have a video that's going to summarize what this is. But complete dominance is an inheritance pattern where the dominant allele always mass expression of the recessive allele. So that's what we've been looking at so far. We've been looking at complete dominance. Therefore, if you see a heterozygous genotype, you will see the dominant phenotype. So that's what we have been doing so far. Incomplete dominance. So we have not looked at any of the characteristics like this, but it's possible to have incomplete dominance. All right. And this heterozygous offspring show intermediate traits. Each genotype then would have its own phenotype. In other words, the phenotype ratio is always the same as the genotype ratio. One example is children born with wavy hair to parents who had both, one had straight hair and one had curly hair. Okay, so what, it's not going to be curly, it's going to be wavy. There's a whole different characteristic. It's wavy. Straight, curly gives you wavy. All right, codominance. Codominance. All right, and we're gonna spend some more time with this because learning about what type of bloods um, when we talk about pedigree, we're going to spend a little more time talking about blood types because that's important. But codominance is a pattern in which the allele is always, both alleles are expressed. Our human blood type is an example of codominance. The table below shows, and everybody should know what their blood type is. All right, so then in this case, there's an allele, there's an A and there's a B. Those are the two types of alleles that you can have. However, if one parent is A and one parent is B, it is possible to have offspring that are AB. Okay. It is possible too that the parents are A, one allele is A and the other allele is O, which has no, it's neither A nor O. I'm sorry, it's neither A nor B, it's O. Okay. In that case, a parent can be AO, they can be BO, and the only time you get an offspring that is completely O is if the parents are donating that O to both. Both parents are donating O. Otherwise, they're going to donate their A, or they're going to donate the B. So there's 4 different blood types that are completely expressed. So we don't just say, oh, it's A and the B is masked. It's AB if they have that other allele. And like I said, we'll look at that one some more later on. Okay, they have it on the chart here. Okay, so those are the different alleles. I mean, these are the different phenotypes, but allele-wise, the parent is type A if they have 2 capital A's or they have an A and an O. In that case, O is like saying nothing. Okay. They're capital B or B and O. And we still say it's type B because the O again is like saying it is nothing. But if they give an A and a B, then the blood type is described as AB. If they get both O's, then the blood type is described as O. Okay. All right. So then we have sex linked. All right. That means that the gene is only found on the X or Y chromosome. Okay, and those are certain genes that have to do with maleness and femaleness. Those genes are only found on the X and Y chromosomes. The other ones can be found on any of the other chromosomes in the body. Okay. All right. And there's a way that you write these. For example, with color blindness is going to, if you have it, they're going to use a lowercase b for color blindness, but it's going to be attached to the X chromosome. So you're going to write X with a B, a little b attached to it. It is not found on the Y chromosome. Okay, so color blindness is not a gene that exists on the Y chromosome. It only shows up on the X, which is why it shows up more in males than females, because if you have an X and you have a Y and there's nothing on the Y to mask what's on the X, it's going to show up. Okay. So this is why there are more male colorblind people than females. For a female to be colorblind, both her X chromosomes have to have that lowercase b, because the other X chromosome could be normal for vision, and it would mask the recessive gene for colorblindness. Okay. All right, so we're going to look at a video real quick. And then we're going to come back to these other ones. Actually, then I might give you time to make sure you get your worksheet turned in. Okay, homepage. All right, now this video will help you as also in because it's set up like a study sheet, like a graphic organizer. So, types of inheritance flow chart.
Hi everyone, this video is to go along with the types of inheritance flow chart. This is a great tool to review before your unit assessment. Just kind of categorize and break down how do each of these 4 types of inheritance compare and contrast. So let's go ahead and first identify what types of inheritance we're looking at. We're looking at simple dominance, incomplete dominance, codominance, and X-linked. Okay, you're going to see some similarities between the first 3 that you aren't going to see on the 4th. So let's go ahead and begin with simple dominance. Okay, simple dominance is autosomal. What that means is it occurs on non-sex chromosomes. Okay, and
vocabulary word, autosomes, non-sex chromosomes. That means all the chromosomes, you have 23 pairs of chromosomes, all of them except for the 23rd one, because this 23rd one is your X and Y chromosomes. It's not on those. Okay, only on the other 22.
In simple dominance, only one allele is totally dominant and overrides another allele known as the recessive allele. So only one allele is dominant and overrides or overshadows the other recessive allele, okay? So for example, capital B is going to be dominant. If you have capital B genotype, it's going to be the dominant phenotype. If you have capital B lowercase b, it's still going to be that dominant phenotype because it's going to override it, okay? And this inherited form is from both parents. Both parents. You get one allele from mom, one allele from dad, and that's how you get your complete genotype, which represents your phenotype. And some examples of this are dimples, freckles, and widow's peak. And the example I gave is also tongue rolling. Okay. Now, the next ones we got into were incomplete and codominance. And I gave you the analogy for incomplete as there is a blend of traits, okay? You cannot be complete without the other one. You are incomplete, you blend. And then codominance where both individuals or both alleles show up to the party. So incomplete dominance is like simple dominance in the way that it is also carried on autosomes. And again, this is a non-sex linked chromosome. Okay. And in terms of the description of this inheritance, what happens is neither of the alleles are dominant, and therefore in heterozygous individuals, a blend of the alleles is observed in the phenotype. So again, there is going to be a blend of traits. If you are homozygous dominant, you're going to show that trait. If you're homozygous recessive, you're going to show that trait. However, if it's heterozygous, it's going to blend. So again, an example of this would be a red flower and a white flower blending together, and the heterozygous one being pink, okay? And again, this is inherited from both parents. And an example of this would be a red flower is big B, a white flower is little b, and a pink flower or a blend is going to be big B, little b. Or you might see this as A, B, and AB, okay?
All right. And why she used B, I don't know, because it should have been R, capital R, lowercase r, because we stick with the color. And then, so it wouldn't have been B because that's random. But the same thing as the example that we just did with straight hair and curly hair. The person would either be all, if they were capital letters, both capital letters, they would have both had straight hair, or they would both recessive letters, they would both have curly hair, or if they had a blend of the two, it would be wavy hair. It wouldn't be curly, it wouldn't be straight, it'd be wavy. So that's the example that they just gave in Canvas.
Now, in codominance, again, this is also an example where you have it carried on autosomes, or again, non-sex-linked chromosomes. And in this type of inheritance, the way I would describe this is both alleles are dominant, and therefore in heterozygous individuals, both alleles are observed directly in that phenotype. So you're not going to get a blend of traits in this one. You are actually going to see both alleles show up at the same time in those heterozygous individuals, okay? So both of the alleles are dominant, and therefore in heterozygous individuals, both alleles are observed directly in the phenotype. And again, this is inherited by both parents. And examples of this would be in blood type. You could have type A, you can have type B, but you can also have AB. Another example of this would be sickle cell anemia, okay? Last but not least, we have X-linked inheritance. And for the first time, this is going to be inherited differently than the other three. And this is inherited on the sex chromosome. More specifically, it's carried predominantly on the X chromosome. Okay? And a description of this would be, it is usually recessive, meaning that females or XX have to have two of those chromosomes to exhibit the trait. However, it can be different, but most of our examples, it's going to have to be homozygous recessive in order for a female to actually get that trait. In males, however, it's only carried on the X chromosome, and therefore they only need one of those recessive alleles in order to inherit that trait. So again, it is usually recessive in nature, meaning that females have to have two affected X chromosomes to exhibit the trait. And that's why males are more likely to inherit X-linked traits due to the fact they only have one X chromosome, okay? And once again, this is inherited. Males get it from mom because they're getting that X chromosome. Females get it from both parents because they're getting an X from mom and an X from dad. So males get X from mom. Females get X from mom and dad, okay? And examples of this are color blindness, hemophilia, and muscular dystrophy. And also we're going to throw this in there, male pattern balding. Okay, so this is a great tool for you guys to review. Okay, I would strongly recommend bookmarking this video and also reviewing this for.
All right, like she said, I strongly recommend that you use this as a graphic organizer. All right. But on blood types, what she left off was there is a fourth blood type. It's O, and technically there are only two. It's an allele. You either have an A allele or a B allele, or you don't have one at all. So there's the O is the absence of an A or a B. So, but you still should have put down that there's blood type O. But blood type O means there's an absence of the A or B allele. So it is, the blood type is O. Okay, I'm type O. All righty, so I'm going to stop sharing, do attendance, mark all those folks late who were late. And then you all make sure that you get that worksheet in. You got about 10 minutes to work on, 10, 15 minutes to work on getting the other worksheet turned in. I'm gonna look right now. I'm gonna tell you who's done it and who hasn't. Like I said, I think y'all took advantage of Thursday. All right, I'll take that back. A couple of you did turn it in. All righty. If you haven't turned it in, turn it in. If you have already turned it in, read over that page, that finish reading that page in Canvas, because it also talked about the other traits. Let me come back over here to my Canvas. All right, so if you're one of the wonderful people who have turned in your worksheets, read actually, let me pull it up again, because we are going to go on to pedigree on Wednesday. And pedigree is going to have another worksheet to do, because you're going to practice pedigree. You're going to practice pedigree and sex-linked traits. All right. So look at these. Do the challenge if you've already turned in, and read it before you go. All right. So again, we're going to talk about pedigree next class. So read that over too, because we're going to have to practice being able to do one of these charts. So that's what we're going to do on Wednesday. We're going to practice doing a pedigree chart, which is different to a Punnett square. Okay, so again, don't sit there doing nothing. Read, 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 read. I'm going to do attendance and I'll be right back. And folks, half of y'all were late today. Y'all got to do better. After 5 minutes, I'm going to start talking, and I'm going to just keep a list of everybody who comes in after the first 5 minutes. Some of y'all came in 10, 15 minutes late. You got to do better than that. That's like showing up just for attendance sake and not getting the work done. I'm not waiting for you. I could have sworn she was here. But she's not here now. And again, if you sign in at the beginning and disappear, then you're absent. All right. Also, the exam that you all have next week is, so the midterm exams start next week in all your classes. For the 5th period exams, and let me pull that up on your page so that you are aware of what I am talking about. All right, on your little video, the slideshow for the week, on the 2nd page, it talks about 9-week exams. You are 5th period, so March the 2nd. March the 2nd is next Monday. So you will have 5th period exam, 1st and 5th period exam on March the 2nd. Your exam will cover everything from January to now, which is cell reproduction and genetics. Most specifically, meiosis and not a little bit about mitosis, but mostly meiosis because that's directly ties into genetics. All the things that we're talking about with the Punnett squares and stuff, that's our conversation about gametes. Meiosis only happens in the gametes. So there'll be more questions about meiosis than mitosis, and there'll be questions about genetics. So again, if you think you're finished and you've turned in everything, don't sit there like and tune out. Start studying. Start pulling out those worksheets. Start figuring out this which question that you don't understand. All right. Just bringing that to your attention. All right. So again, if you've turned in your worksheets, read. If you haven't, or study. If you haven't turned those worksheets in, and I got a couple of you that are missing both worksheets, And I'm running into a lot of people who are getting this order of things incorrect. So let me pull that up real quick. And okay. Y'all did what you wanted to do with these worksheets. Actually, I'll pull it up on Wednesday. Y'all keep working. I hope so. Stop moving. Oh my goodness. My pen is losing its mind. Christopher is not here today. Okay, folks. If you haven't finished up, please make sure you finish up because that was due today. Wednesday, like I said, we're going to, I'm gonna go over these because a lot of you got the sentences wrong on the genetics, the first genetics worksheet. So we'll go over that and then we'll talk about how to do a pedigree chart. Okay. All right. So have a great afternoon. Stay warm. It's cold. It's going to be cold again tomorrow.